Hello dear friends, welcome back again to another video from my YouTube channel. Dear friends, in our previous video we talked about the artificial hybridization process. In this video we are going to understand about one more very interesting process, that is pollen-pistil interaction. The textual meaning of this entire concept is there is an interaction between pollen grain and a pistil. And what kind of interaction it is? Interaction means there is a communication, identification. What kind of identification? What kind of communication? What kind of interaction between pollen grain and a pistil? There is a chemical interaction between pollen grain and a pistil to recognize the compatible pollen grains or to recognize the right type of pollen grains. That is collectively called as pollen-pistil interaction, and this concept is very interesting. Let us try to understand the pollen-pistil interaction in detail. As we have discussed earlier, there is a chemical identification, there is a chemical interaction, there is a chemical communication between pollen grain and a pistil. What here pistil refers to? Pistil in this concept refers to the stigma, because stigma is the most important part of the gynoecium, and this single individual unit of the gynoecium, it is collectively called as pistil. And the stigma being a most important part, it receives the pollen grains after pollination. In nature, there are so many pollen grains. There are released in the environment. Pollination not always guarantee the deposition of right type of pollen grain on the stigma. Say for example, this gynoecium belongs to the mango. In nature, we don't have only the pollen grains of mangoes. We have the pollen grains of neem also. We have the pollen grains of acacia also. We have the pollen grains of Tamarindus indica also. We have the pollen grains of various plants. They are simply there in the environment. That means pollination not always guarantee the deposition of right type of pollen grain on the stigma. There may be the pollen grains of mango landed on the stigma of neem. There may be a pollen grain of neem landed on the stigma of the mango. This may happen in the environment. This is happening in the environment. So, how to recognize the right type of pollen grain and how to reject the wrong type of pollen grain? That entire concept is studied under the heading pollen-pistil interaction. Before we discuss this, let us make a very clear statement that each and every plant consisting of a specific type of chemicals in it. Mango plant has got a particular chemical in it. Neem plant has got a particular chemical in it. Acacia nilotica plant has got a particular chemical in it. Maize plant has got a particular chemical in it. Sorghum bicolor has got a specific kind of chemical in it. That means each and every plant in the nature consisting of a specific type of chemicals, and these chemicals, they help during the pollen-pistil interaction so that the stigma can recognize the right type of pollen grain and stigma can reject the wrong type of pollen grain. Let us understand in detail with the help of this diagram, my dear friends. Scientist has defined the pollen-pistil interaction as all the events from pollen deposition on stigma, all the series of events from pollen deposition on stigma until until pollen tube enters into the ovule is called pollen pistil interaction. That means the series of events happening from the deposition of pollen grains on the stigma until the pollen tube enters into the ovule. What is this pollen tube? We'll learn little later. Pollen tube. What is this pollen tube and which cell in the pollen grain produces the pollen tube? We'll discuss in detail here. This is the standardized definition of pollen-pistil interaction, my dear friends. Now, let us try to analyze what are the events, what are the series of events happening during the pollen-pistil interaction. Pollination occurred and there is a deposition of two types of pollen grains. That means there is a landing of two type pollen grain on the stigmatic surface here. Now, consider this gynoecium or this flower, this female part. It belongs to the mango, for example. Mango. It is the mango pistil. On the stigmatic surface of the mango pistil, there are two pollen grains landed. Consider this pollen grain belongs to the mango. This belongs to the mango, for example. Mango pollen grain is landed on the stigma of the mango plant. Here, consider this pollen grain belongs to the neem plant. Neem plant. This may happen in the nature because the pollen grains are simply floating in the air. Any kind of pollen grain may land on the stigma. Any kind of pollen grain. Here, the two pollen grains are landed on the stigma. One belongs to the neem plant and the other belongs to the mango plant, but the stigma belongs to the mango plant. In these two conditions, in these two situations, there is a chemical communication, there is a chemical identification between the pollen grain and a stigma. This entire thing is the stigma. Stigma has a capacity to select the right type of pollen grain and stigma has a capacity to reject the wrong type of pollen grain by matching their chemicals. Just now I told that each and every plant consisting of a specific type of chemicals in it. That means mango pollen grain, it consists of, for example, XYZ chemical. Similarly, the stigma of the mango, it is also consisting of XYZ chemical. Here we have got XYZ because mango, it is consisting of XYZ. There is no such XYZ chemical in the biology. We take this XYZ as an example. Mango plant, for example, consisting of XYZ chemical. That means pollen grain of the mango plant, it is also consisting of XYZ chemical. But the neem plant, for example, it consisting of ABC chemical. That means the pollen grains of the neem plant, they consist of ABC. Here, as the pollen grains, they landed on the stigma, suddenly stigma recognizes the right type of pollen grain by matching their respective chemical. Here we have got XYZ chemical. Here also we have the XYZ chemical in the stigma. Here we have XYZ chemical in the stigma. Here we have got ABC chemical. That means here there is no chemical matching. Here there is a chemical matching. This chemical matching is only to select the right type of pollen grain and to reject the wrong type of pollen grain. And the right type of pollen grain is called as compatible pollen grains. Compatible pollen grains. And the wrong type of pollen grains are called as incompatible pollen grains. Now in these two situations, where is the compatibility and where is the incompatibility? Here there is a compatibility, here there is no compatibility. Hence the stigma rejects this pollen grain and stigma selects this pollen grain. This is the first step. This is the first event occurred during pollen-pistil interaction. What is the first event? There is a chemical interaction. There is a chemical interaction between pollen and pistil to select compatible pollen grains, compatible pollens, and to reject incompatible pollens. This statement clearly tells us that stigma has a capacity to select and to reject. Who has the capacity? Stigma has a capacity, and that capacity is due to the chemical matching. Here there is a compatibility and here there is an incompatibility. Where there is a compatibility, stigma start secreting. Here there is a compatibility, here there is no compatibility. Stigma starts secreting the sugary substance at the base of right type of pollen grains. Here it is sucrose, actually sucrose, sugar secreted at the base of compatible pollen grains. Here there is no compatibility, that means here there is no secretion of the sucrose. At the base of compatible pollen grain, stigma secrete sugary material or substance. Stigma secretes the sugary material. Whereas at the base of incompatible pollen grain, there is no such secretion. Now this sucrose enters into the pollen grain. It enters into the pollen grain through the germ pore. What are the germ pores? They are the openings in the exine of a pollen grain. They are called as germ pores. Sucrose get entered into the pollen grain through the germ pores that stimulates the vegetative cell. What is that vegetative cell? Recall the structure of the pollen grain, my dear friends. Vegetative cell is a nourishing cell. Here rich amount of nutritive materials get stored in that cell. That is why it is swollen and the bulgy cell. Sucrose enters into the pollen grain and that stimulates the vegetative cell, and vegetative cell dissolves and starts producing a tube. Here. Which cell produces the tube? Vegetative cell produces the tube after getting stimulation by this secretion secreted by the stigmatic surface. What is the next event? Stigmatic stigmatic secretion, that is nothing but a sucrose, stigmatic secretion stimulate the vegetative cell of pollen grain to produce a tube called pollen tube. Pollen tube. This event is called as pollen germination. This event of production or this event of forming a pollen tube on the stigmatic surface, it is called as pollen germination process. Now the pollen tube enter into the stigmatic surface. Simultaneously, there is one more cell in the pollen grain, that is the generative cell. Both vegetative cell and generative cell, they are haploid. This generative cell divides by mitosis and results into the formation of two haploid male gametes. Two haploid male gametes are formed. And these two haploid male gametes, they remain attached to one another. They lie very close to one another after the division. Here, vegetative cell produces the pollen tube after getting stimulated by the stigmatic secretion. Simultaneously, generative cell undergoes one mitotic division and results into the formation of, now it is no more called as generative cell, they are two male gametes. Two male gametes are formed. And these two male gametes, they follow the pollen tube. Now these two male gametes are here. They follow the pollen tube. As we have discussed earlier, generative cell is rich in its nutrient, and that nutrition, now it is being utilized for the growth of this pollen tube. Now the pollen tube is growing through the style. First it entered into the stigmatic surface. Now it is growing through the style towards the ovary. Pollen tube gets secreted. Pollen tube gets formed on the stigmatic surface. It entered into the stigmatic surface. Now the pollen tube is growing through the stylar region and it is growing towards the ovary. Why it is growing towards the ovary? Because the female gamete is located within the ovule of the ovary. That is the main intention of growing this pollen tube towards the ovule of an ovary. Now it has reached the ovary. Let us write the point here. Pollen tube germinated or pollen tube formed on stigma, enter through the style and growing towards ovary. Now, when the pollen tube reaches the ovary, there are three possible ways of entering this ovule. There are three possible routes of entering this ovule. The first possible route, it may directly enter, that I am mentioning in the form of dots. The first possibility of entering this pollen tube into the ovule is through the, like this it may enter, like this it may enter. This is the first possibility. Second possibility, it may enter like this. It may enter like this. This is the second possibility. The third possibility is it may enter like this. It may enter like this. This is the third possibility. Let us recall the three possible ways of entering the pollen tube into the ovule. The first possible way of entering the pollen tube into the ovule is through the, which end it is? Is it a chalazal end or micropylar end? It is the chalazal end. Here we have a mouth kind of thing. It is a micropylar end. This is the micropylar end and this is supposed to be the chalazal end. The first possible way of entering the pollen tube into the ovule of an ovary is through the chalaza. If the pollen tube enters the ovule through the chalaza, pollen tube entry through chalaza. This is the first possibility. Second possibility, the pollen tube enters through the integuments, outer and the inner integuments, protective layers of the ovules. This is the second possibility. Pollen tube entry through integuments. Then the third possible way of entering the ovule is through the micropylar end. Third possible way is pollen tube entry through micropyle. This entry are given with the particular technical terms. If the pollen tube enter the ovule through the chalaza, then it is called as chalazogamy. What is chalazogamy? The entry of pollen tube into the ovule through the chalaza and it is called as the chalazogamy. If the pollen tube enters the ovule through the integuments, then it is called as mesogamy. If the pollen tubes they enter into the ovule through the micropylar end, then it is called as porogamy. These three are very, very important for competitive exams, my dear friends. Chalazogamy means pollen tube entry into the ovule through the chalazal end. The entry of pollen tube into the ovule through the integuments, it is called as mesogamy. If the pollen tube enters into the ovule through the micropylar region, then it is called as porogamy. Out of these three types of entry, porogamy is the most common type of entry in almost all the angiosperms. Why it is most common type of entry? Because if the pollen tube want to enter the ovule through the chalaza, it has to break the integuments. If the pollen tube want to enter into the ovule, it has to break the two integuments here. Here there is an opening, there is no integuments, it's a clear-cut entry here. It is a very easy entry for a pollen tube to enter into the ovule. They are the three possible ways. There are the three possible ways of entry. That means this entry, let us make it darker. This entry is the very common type of entry. This is very common type of entry of a pollen tube into the ovule. Now, these two male gametes, they are carried here now. They are carried here. These two are the male gametes. They are carried here. As the pollen tube reaches the micropylar end, we have the synergids here. Synergidal cells. Inside the synergidal cells, there are some finger-like projections. These finger-like projections are nothing but filiform apparatus. The filiform apparatus guides the pollen tube into the ovule. This filiform apparatus, it guides the pollen tube to enter into the ovule. Now, there is a complete entry of the pollen tube into the ovule. As there is an entry, these two male gametes are here now. These two male gametes are here. That means the pollen tube has entered into the ovule through one of the synergid. This synergid helps, or this filiform apparatus of the synergidal cell guides the pollen tube so that the pollen tube can enter into the ovule. As it was guided, suddenly this synergid cell start getting degenerated. This start getting degenerated. Why? Because the pollen tube made its entry into the ovule through this synergidal cell. That means out of two synergids, one synergid degenerates during the entry of pollen tube. Who guides the pollen tube entry into the ovule? It is filiform apparatus. Let us write that point. It is very, very important. Filiform apparatus guides. Filiform apparatus guides the pollen tube during entry into the ovule. Up till now, what we have seen is the pollen grain is landed, the pollen grain is recognized, the right type of pollen grain is facilitated with a sugary substance. Then the pollen grain start forming a pollen tube. Pollen tube entered into the ovule. What is going to happen next? That we are going to study in our next video under the heading of double fertilization process. I hope you people have understood this entire complicated. I made it very simple for you people. It's very interesting process. In our next video, we're going to discuss what is going to happen next after the entry of this pollen tube into the ovule. Till then, stay home, stay safe. Thank you so much.